This topic is about application of plant pisciculture in agriculture and horticulture. I am going to talking about introduction. Plant pisciculture is an essential component of plants biotechnology. The possibility to regenerate whole plants from protoplasm, single cells, tissues, and organs in vitro has opened out entirely new approaches to plant improvement. Agriculture was the key development in human civilization where farming made humans stay, occupy, and build at place. It feeds a large population of people without any limitations, but over the year, the growing population and shrinking space made it is difficult to fulfill the demand for plant food. Next one is the application of PTC in agriculture. First one is production of rare hybrids. Hybridization is a well established plant breeding procedure to obtain superior plants by combining useful characters distributed in different plants. Embryo culture has been successfully applied to several sexually incompatible crosses. In several interspecific crosses of brassica, abortion of a hybrid embryo occurs at such an early stages that it is not possible to exercise and culture the embryo. A hybrid can be obtained by culture the ovule culture, including the hybrid embryo. Embryo culture has been used to raise hybrids between the sexually incompatible parents, Gosbium hirsutum. Next one is application of different tissue culture technique in crop improvement. First one is somoclonal variation. The variation in plant cells is the result of the several events including environmental, genetic, and epigenetic events. But tissue culture has also been essential to induce somoclonal variation. Next one is micro propagation of plants. Micropropagation is a popular technique to propagate plants on a commercial scale. It has been popular for more than 30 years in labs and industries for the production of plants for different purposes. The technique of enhancing axillary bud breaking is the most commonly used technique for commercial micropropagation. Next one is somatic hybridization and hybridization. Somatic hybridization involves the fusion of somatic cells, regeneration of plants from the fusion product. Plant cells are bounded by a rigid cellulose walls are cemented together by a pectin-rich matrix to form tissues. An essential step in the fusion of plant cell is to bring together the plasma membrane by degrading the cellulosic wall. Thus, the first step in somatic hybridization is the isolation of plant protoplast. Next one is synthetic seeds. A synthetic seed is a somatic embryo encapsulated inside a coating. It's of different types, which include somatic embryos encapsulated in a water gel, dried and coated somatic embryo. Somatic embryos are suspended in a fluid carrier. Shoot buds encapsulated in a water gel. Next one is haploid production. Haploids are extremely important in genetics plant breeding. In haploids, it is possible to detect recessive mutant which do not express themselves in the diploid state due to the presence of dominant allele in cross-pollinated plant. F1 hybrids with a high degree of heterozygosity, the fixation of a particular trait through the conventional method of back crossing takes 7 to 8 years. Next one is application of PTC in horticulture. Clonal propagation. The conventional method of clonal propagation are slow, often not applicable. For example, the only in vivo method for clonal multiplication of cultivated orchids which are complex hybrids is back-bulb propagation. It involves separating the oldest pseudobulb to force the development, but this process allows at best doubling the plant number and every year. In vitro clonal propagation, popularly called micropropagation. Micropropagation generally involves 3 steps. Next one is germplasm preservation. The germplasm preservation is an in vitro storage of plant genetics under slow growth conditions of cryopreservation or as desiccated synthetic seed. It's alternative to seeds banks and fields collections of clonally propagated crops. Lack of a common method suitable for all species and genotypes, high cost, the possibility of somoclonal variation. Next one is shoot multiplication. The most popular method of shoot multiplication is forced proliferation of axillary shoots. For these cultures are initiated from apical or nodal cutting carrying one or more vegetative buds. The presence of cytokinin alone or in combination with a low concentration of an axine such as indolestic acid, naphthalene, acylic acid, the pre-existing buds grow, produce 4 to 6 shoots within 3 to 4 weeks. And second is rooting. Shoots are produced through axillary branching or adventitious differentiation are rooted in vitro on a medium containing a suitable auxin such as IANA, IBA. Alternatively, where possible, the shoots are treated with auxin directly planted in the potting mixture for in vivo fruiting. Next one is transplantation. The shoots are plantlets multiplied on a medium containing organic nutrients, so poor photosynthetic capability. In practice, the plants are maintained under high humidity for 10 to 15 days after they removed from culture results. During the next few weeks, the humidity around the plants is gradually lowered before they are transferred to natural conditions. The two main applications of plant tissue culture in horticultural production are in propagation and sanitation. Tissue culture is being used effectively in asexual clonal propagation of various horticultural crops, orchids, carnations, roses, gerbera, chrysanthemums, potatoes, strawberries, apples, and coffees have all been successfully cloned in vitro, and list is increasing daily. Recovery of pathogen-free plants by meristem tip culture has been become a common practice in producing virus-free stock material of vegetatively propagated plants. Other areas where tissue culture can be contributed horticulture include embryo culture and germplasm preservation and storage. Next one is process of disease-free plants. Under normal conditions, plants are infected by a wide range of pathogens such as bacteria, fungi, virus, viroids, insects like nematodes and insects. If plants for micropropagation are derived from an infected plant, the pathogens can multiply, spread to a large number of plants. Eradication of virus and other pathogens is also desirable from the point of view of international exchange of plant materials. Thank you.